in the field around the world. This is the outline of my talk. We will have a brief background and then followed by the system introduction and a few case studies, a quick summary, conclusion, and then open for any questions you may have. So this is a simple demo of how heart beats. Action potentials start at assay node, and then the action potentials are conducted through every node and Purkinje fiber and his bundle to reach all myocytes. And then myocytes contract together and heartbeat. And the surface ECG is the ensemble of all electrical activities. For action potential of each cardiomyocyte, the different phase of action potential is contributed by different cardiac ion channels. For example, at phase 0 is the sodium 1.5 channel open, which depolarizes the membrane potential and allow L-type calcium channel to open. And then the action potential go to plateau phase. During phase 2, small amount of extracellular calcium enter cardiomyocyte via this L-type calcium channel, activates renin receptor and leads to large amount of calcium released from internal SR storage. Those calciums then bind to troponin C to activate the myofilaments and cause muscle contraction. So this is so-called the EC coupling. And then the K channel, mainly is the hERG and iTest channel, are activated to repolarize the membrane potential. To bring the membrane potential back to resting state, and then the calcium is disassociated from troponin C and pumped back to SR and extracellular space, resulting in muscle relaxation. So usually the action potential duration at 90% high, or they call it, or we usually call it the ATD90. In action potential duration is well correlated to QT duration in the ECG signal. And off-target effects on cardiac ion channels to cause alteration of action potential shape and duration, sometimes or cause the earlier after depolarization of the EAD, resulting in abnormal surface ECG. So for example, if you block sodium 1.5 channel, the result is the QRS prolongation could lead to every block. Actually, it's a ventricular arrhythmia. If you block the calcium 1.2 channel, and then it could cause PR prolongation. This one could cause every block. Blockade of a hERG or IKS channel is a well-known effect of the QT prolongation, and a severe case could lead to a torsade point, which is fatal ventricular arrhythmia. Blockade of IK1 or the KR2.1 channel can also lead to QT prolongation and arrhythmia. So why we focus on cardiovascular safety? The reason is very simple. Cardiotoxicity contributes to about 40% toxicity related drug failure or withdrawals. Drug development of many small molecules was halted due to off-target effects on cardiac ion channels and other cardiac safety concerns. Among these, hERG has become the center of the role, and hERG channel counter screen play a critical role in early stage drug development. Recently, all those withdrawing drugs are all potent hERG channel blockers. Here is a non-exhaustive list. So, hERG channel is thus the major target for drug-induced QT prolongation. And currently, the ICH S7B guideline recommends an in vitro hERG evaluation of all pharmaceutical compounds that are targeted for human use. However, I want to highlight that not all hERG blockers can cause QT prolongation, and not all QT prolongation can lead to a torsade point. So, in June 2013, FDA hosted a workshop aiming to change in ICH E14 and S7B guidelines for better assessment of drug-associated proarrhythmia. plan to revise ICH S70 guideline by July 2016 to include studies on other cardiac ion channels in addition to HERG, and to include studies use stem cell-derived cardiomyocyte. Also plan to revise to remove the ICH E14 guideline because the solar QT study was very costly, and yet the outcome is not very specific to evaluate drug-related or drug-induced ventricular proarrhythmia risk, because there are a lot of non-proarrhythmic factors such as glucose levels, body temperature, or even circadian rhythm and prolonged QT for those participants in the studies. So what's CiPA? CiPA stands for Comprehensive In Vitro Proarrhythmic Assay. It's an initiative responding to FDA's call for action from the workshop. And the purpose of the CiPA is to evaluate direct proarrhythmic risk of test compounds. So the highlight I want to highlight is to evaluate direct effect. And the proposed measure is to conduct multiple cardiac ion channels and then integrate those data into a computer model, and then verify the modeling result with stem cell-derived cardiomyocyte to have a better assessment of this proarrhythmic risk of the test compounds. So then the question is how to conduct the studies and how to implement the initiative. And one part of a proposal is to measure field potential of this stem cell-derived cardiomyocyte. This is a classic single-cell patch clamp recording to measure action potential from either stem cell-derived cardiomyocyte or even from a fresh isolated cardiomyocyte. And this is the heart, and this is the surface ECG signal, which is the ensemble of all individual action potentials. And this is a synthesis of a stem cell-derived cardiomyocyte that can be together. The electrical signal from the synthesis measured with electrodes is the field potential. It's the ensemble membrane activity of all cardiomyocytes in the synthesis. And there are good linear relationships between rising time of field potential and X potential, and the duration of field potential and X potential. The T wave is reflected by the field potential max. So I want to bring attention to this so-called T wave in the field potential. In real case, the T is not as obvious as shown here. This is a picture of the ACEA CardioECR system. The CardioECR system monitors cardiomyocyte synthesis beats in a 48-well plate format. Cells in close contact with gold electrodes at the bottom of each well, and the impedance signal was generated by applied low voltage creating current between electrodes. And there are two field potential electrodes in close proximity to synthesis in each well. It's a simultaneous real-time measurement of synthesis contractility by impedance and electrical activity by field potential measurement. It's a high throughput system. The endpoints for impedance are the cell index, which is the baseline impedance reflecting synthesis attachment and also the cell health. And then the bit rate, which is the number of both cycles between the beginning of each of first detect cycle to the end of the last detected cycle during the 1 minute recording time. And then the beat amplitude. The endpoints for field potentials we use are the field potential spike amplitude, the field potential rate, and the field potential duration. So the main advantage of a CardioECR system is that it's a non-invasive, continuous recording in cell incubated. It can last for days. And it's a simultaneous and multi-parameter analysis of a stem cell-derived cardiomyocyte in vitro. And from the impedance parameters, the cell index can indicate cell attachment and cell health or cytotoxicity. And the impedance can provide cell contractility data. From the field potential parameter, is ensemble data of a stem cell derived cardiomyocyte membrane electrical activities, and it's an integrated readout of drug effect on all cardiac ion channels expressed in those stem cells. And also, the impedance signal can help to identify and understand field potential signals. For example, the detection of T wave. I will talk in more detail for this one in the following slides. And the combination of impedance and field potential signals can help to correctly understand test compound effects. In most cases, it applies to have multiple parameters for test compound effects. And in some cases, it's actually a must to correctly explain test compound effects. For example, whether it's a cytotoxicity or it's a test compound effect that lead to attenuation or disappear of field potential spike. Is the EC decoupling or is a conduction block in sensation of impedance signal? So we conduct a pilot study to evaluate test compound effects on the stem cell-derived cardiomyocyte with field potential measurement using this ACEA CardioECR system. The data analysis software, the RTCA CardioECR software, is fully functional and can quickly and reliably detect and export field potential spike, amplitude, firing interval, and rate. And the arrhythmia can be easily identified by visual inspection of the spectrum. However, the detection and identification of the so-called T wave in field potential by the software is still a challenge. Therefore, it's currently handled manually using the software as this. First, the user visually inspects and corrects, if needed, the detection of the T wave, and then the software will calculate the corresponding field potential duration. So first, we study mechanistic fluxing. This is the cell index spectrum. The red line is at the time the compound was added to the well. You can see after the addition of the mechanistic fluxing, the cell index after normalized to the green rate control is very stable. So a stable cell index after the compound addition indicates its non-cytotoxicity. And this is spectrum before the read, or it's like control spectrum. This part is the impedance signal. That's how it looks like. It's a whole 1 minute spectrum. This is the field potential signal, how it looks like. So 30 minutes after the addition of mechanistic fluxing at different concentrations from 3, 10, 30 to 100 micromole. This is the result. I zoom this further and align these two signals together. The field potential signal and the impedance signal can be well aligned. And here you can see, in the presence of mechanistic fluxing, the impedance and field potential signal was mainly slowed down, but we did not see any EAD up to 100 micromole in the presence of mechanistic fluxing. So then we studied flattening. Again, this is a cell index, and the stable index tells me that the flattening won't kill the cell. So control freely, and 30 minutes later, with different concentrations of flatteners from 100 nanomole to 3 micromole. And visually you can tell the drug clearly slowed down the rate, and both the rate is slowed down. But I want to bring your attention to the amplitude. So in that case, the impedance amplitude was not very sensitive to sodium channel blocker flattener, while the field potential amplitude clearly demonstrates test compound effects in a concentration dependent manner. I zoom this spectrum further. This is the freely and this is after 3 micromole platinum addition. And both spectrum has the same time scale and absolute value scale. So here you can see very clear that impedance amplitude was reduced only about 40% by 3 micromole platinum, but the field potential amplitude reduced more than 90% in the presence of 3 micromole platinum. So from this case study, we learned that if only based on impedance signal, the test compound effect mainly say on the sodium channel will be underestimated. Next, we study mechanistic. So this is the control, and then 30 minutes later after compound addition. And we test 1, 3, 10, and 30 micromole. So compared to platinum, mechanistic demonstrate a different case. The amplitude of both impedance and field potential was down to limit of detection with 30 micromole mechanistic. So this effect we interpreted as the synthesis start beating at that concentration. And next we start cleaning, and this is a full 1 minute spectrum in the presence of cleaning. And we can use the RTCA ECR software to average the spectrum. This is the average of all field potential weight within this 1 minute. So now comes the question, where is the T? Is this the T wave, or this one, or this little one? If just by eye, it will be difficult to judge. So what we do is, how about we superimpose the field potential signal with impedance signal? This is the spectrum. We superimpose the average of both signals in the presence of our clinic. When impedance signal was superimposed to field potential signal, it becomes clear that the inflection point we picked in the previous slide at the last arrow point is well aligned with the inflection point in impedance signal. Therefore, we think this one is the T wave in the field potential signal. So, in the case of a candidate, we can use this detection method to detect the QT prolongation of the drug effect. And the average field potential waveform of before and after cleaning addition was superimposed here. The green trace is before, and the red trace is after. To use the criteria detection method I described above, we can detect the T wave pre and after, and 30 minutes after 300 nanomole cleaning addition. Here you can see a clear prolongation in this field potential duration. It's a circuit of the QT prolongation by 300 nanomole cleaning. In the study of cleaning, we also observe EAD. This is a prelude of the field potential signal. And in the presence, 30 minutes later, after 1 micromole, so the concentration is higher than the just QT prolongation. This is 1 micromole cleaning. This is expression. First, is very obviously is the amplitude drop, and the duration or the interval of the field potential was prolonged greatly. It's almost duplicate. And then this, a circle with a green circle. This is the EAD, or earlier after depolarization. It's a arrhythmia. So next, we study calcium channel blocker, nifedipine. And again, the prelude and the 70 minutes later, after compound addition, we test 10, 30 nanomole and up to 300 nanomole of nifedipine. This one is different. So nifedipine, we can see that its concentration dependently increased beating rate and reduced impedance amplitude, while the field potential amplitude was not very sensitive to this calcium channel blocker. Compared to the flecainide, it's very sensitive to sodium channel blocker flecainide. And then as nifedipine made the cell speak faster, so we have a chance to see whether it constitutes shortening. So we superimpose the average spectrum of still potential before and after spectrum together. Again, the green one is the before compound addition. The red trace after nifedipine addition. Use the detection method. We detect the T rate is the free rate, and this is 30 minutes after 10 nanomole nifedipine addition. In this case, the QT, or actually is the still potential duration, was shortened by about 20% by 10 nanomole nifedipine. So this is the summary of the 8 compounds we studied for this CiPA pilot studies. For the interest of time, I won't go through all the data. I just want to mention that in general, the study result was well aligned with literature report and with our own internal studies with other platforms. So in conclusion, I hope I can mention you that data generated in the pilot study with all 8 well-known standards was in line with literature and being satisfactory. Compared to other MEA systems, a major feature of CardioECR system is to simultaneously record syncytial contractility by impedance and electric activity by field potential. And multiple parameters provide advantage in evaluating test compound effects as shown in those case studies. And we can cross-reference impedance data to identify T rates and field potential. The cell index data can be used to exclude cytotoxicity. The field potential amplitude is more sensitive to sodium channel blocker, while the impedance amplitude is more sensitive to calcium 1.2 channel blocker. And when both signals disappear, we can believe it's a true sensation of syncytial beating. So during the pilot study, we realized that the field potential amplitude had relatively large variations. Therefore, we conduct a time course study with vehicle control, which is a 0.1% DMSO, for 2 days, continuous recording in incubator for 2 days. And the field potential rate and field potential amplitude was normalizing to clearly. That's the final read before this vehicle addition. And the red plot here is the field potential amplitude. And the blue plot here is the field potential firing rate. I want to bring your attention to the relatively large error bar by the field potential amplitude. In comparison, the error bar in the field potential rate is very small, barely visible here in this plot. And the sampling rate we use is 10 kHz, or is like a 0.1 millisecond, which is high enough to capture any sodium channel signal. Therefore, the variation in this sodium spike for the amplitude of the field potential amplitude should not be due to undersampling rate. So we think about this, and we think one is more like a speculation, but we think one possible cause could be a small change in the distance at the contact between the cell, and the recording electrodes. Of course, this is an open, just an open speculation. But we do observe a similar phenomena with other MEA systems. So I hope this can bring just like an open question for people to discuss. And thank you for your attention. I will be more than happy to answer any questions you may